So I'm a professor at the University of Alberta, working mainly in prostate cancer for the past 15 years or so. Launched several companies based on the research that we developed in the lab. These are my disclosures, and I'll be talking a lot about what some of these companies are doing today as well, so it's relevant. All right, so obviously, cancer is a huge societal burden, health burden, and certainly as we get older, we have an increased chance of getting cancer. But the way we treat cancer has changed significantly over the past few decades. While cancer diagnosis may have been felt like a death sentence 50 years ago, now more and more cancers are becoming livable, more longer-term diseases. However, for the majority of cancers, we still have treatments that I would say are relatively archaic and lack what I'm going to be talking about a lot today is selectivity. So treatments like traditional chemotherapies, radiation, surgery, have a long track record of improving outcomes, but they also cause pretty significant morbidity because they lack selectivity for cancer cells specifically. Chemotherapy is a great example. Many of the traditional chemotherapies target all dividing cells in the body, and obviously cancers are highly metabolic and they're dividing, and so these treatments selectively kill them to some degree, but they also selectively kill many other cells in the body, the lining of the gastrointestinal tract and hair follicles, etc. This can cause pretty significant side effects, and some of these side effects can persist, for the rest of the patient's life. We thought with the advent of just amazing ability now to sequence tumors, to understand the underlying genetics of tumors, to understand the underlying genetics of patients, which can really drive the natural history of their tumors, we've seen a huge opportunity to really apply these new and exciting genetic medicine approaches to the treatment of cancer. And we're seeing this through gene therapies, through really interesting, siRNA and RNA inhibition therapies in cancer, and now obviously through ex vivo cell therapies like CAR T. But I think the goal that I see that's really attainable now with technologies that we have are to develop, dosable, specific gene therapies that can be given sort of to any patient. And while we have pretty amazing tools now to be able to upregulate and downregulate genes expression, to be able to even edit genes, what we've lacked, I think, and what we really need is a safe, effective, redosable nucleic acid delivery technology. And so I spent a lot of time in my lab and several companies really trying to work on this issue of being able to have a redosable genetic medicine technology. And so if you look at sort of what's available commercially now, either in the gene therapy space or the non-viral therapy space, we really have the option of viral and non-viral approaches. On the viral side, we have adeno-associated viruses, probably the most common and commercialized platform. And obviously, viruses have evolved for millions of years to be able to very effectively, penetrate tissue and deliver the genetic material, typically DNA, intracellularly for the purpose of replicating the virus. And but of course, we've hijacked this machinery to deliver, therapeutic genes. And so AAV has been very successful in very specific circumstances. But we're finding if we're taking out AAV and trying to apply it more broadly to gene therapy, we're seeing some pretty significant issues. First of all, tolerability of very large systemic doses required. Many of these genetic diseases, we're seeing tolerability challenges, even some deaths in patients. Of course, it's a virus, so we have immunogenicity. So your immune system induces an immunogenic response against the virus, and that really limits or completely prevents the ability to repeat dose. And of course, viruses are designed to deliver DNA with a certain cargo capacity. And so non-viral approaches like lipid nanoparticles were really developed to address a lot of these limitations. Unfortunately, I think for systemic administration, LNP have had a significant roadblock, and this is basically related to their mechanism of action. They utilize potent ionizable lipids that upon endocytosis and acidification of the endosome sort of change their shape, acquire a charge, sort of break open the endosome, And this induces, and because they're relatively tropic to tissues like liver, this has induced some challenges trying to hit extrahepatic targets like tumors, for instance. And so we're seeing these tolerability issues trying to hit extrahepatic targets at high doses. Of course, they're not immunogenic, so repeat dosing is possible. And they're able to deliver very effectively RNA and siRNA, for instance. And so, we thought, what if we could combine sort of the best aspects of viral and non-viral delivery? So we've done that with a platform that we call the Fusogenics Proteolipid Vehicle, or PLV platform. Basically, what we've done is we've taken a really interesting, novel, tiny viral fusion protein. And what's really interesting about the activity of this protein is it's able to potentiate direct fusion of a lipid particle with the plasma membrane of a target cell, basically inducing direct fusion and dumping the cargo into the cytoplasm. And so we've, when we do this, we've been able to sort of reimagine the lipid components of a lipid particle to create sort of the platform that can deliver either RNA or DNA, and it can be administered repeatedly. And I'll get a little bit into detail. So obviously viruses have evolved over millions of years to be able to pretty effectively enter target cells. Envelope viruses in particular express a wide variety of different fusion proteins on their envelope. You can see that it's sort of divided mainly into three classes here. And viruses like HIV, like flu, all have these fusion proteins that basically perform a very similar function, although their structures can be quite different. They bring opposing lipid membrane bilayers together. They create an environment where lipids can mix between the two layers, eventually forming a pore, stabilizing that pore, and expanding it to create a full fusion event. But if you look at these fusion proteins, they're typically pretty huge. So they have these pretty massive ectodomains. They're multi-subunit, so they can be immunogenic themselves. From a manufacturing standpoint, pretty challenging, I think, to manufacture and incorporate a drug delivery system, which is why, we were very fortunate. Dr. Roy Duncan, he's a virologist at Dalhousie University and a co-founder of Ventus Pharmaceuticals, discovered some almost 25 years ago, the only non-envelope virus to express a fusion protein. And so this fusogenic orthoreovirus actually doesn't affect humans. It affects things like alligators and bats. And what's really interesting is that it's not envelope, so it doesn't use a fusion protein to enter cells. But once it enters cells, it very quickly expresses a fusion protein and causes them to fuse with each other in these huge multinucleates in situ. Yeah, so again, what's interesting about this fusion protein produced by this fusogenic orthoreovirus is that it's evolved completely separately than these envelope virus fusion proteins. And as a result, it's super tiny. So it's got a very, very tiny ectodomain, just a dozen amino acids. It's a single transmembrane domain. Roy called these the fusion-associated small transmembrane, or FAST proteins. And what he showed back in 2005, in this seminal paper in EMBO journal, is basically he incorporates these FAST proteins into a lipid particle with labeled lipids and a labeled cargo and incubates them with target cells. He found that very, very quickly, he was able to see content mixing with the cargo of the lipid particle and the cytoplasm of the target cell, mixing up the lipids with the plasma membrane of the target cell. What was actually pretty exciting, looking at the mechanism of delivery, when he incorporated a fusion-inhibiting lipid, lysophosphatidylcholine, he was able to see a really nice dose-dependent reduction in this content of lipid mixing. But, and this is the most interesting, when he inhibited endocytosis with a variety of different endocytosis inhibitors, basically was only able to inhibit the delivery by some 17 or 18%. And so this seems to be a completely endosomal escape independent mechanism of delivery. And so, the FAST protein story is a little bit more complicated than one fusion protein. Turns out there are about 6 different fusogenic orthoreoviruses that express FAST proteins. So Roy and his lab spent better part of 2 decades discovering these and characterizing them, and he and I in our labs have worked together to do structure function analysis to sort of figure out what gives them their activity. And this is just an example of an experiment where we basically took 2 of the parental FAST proteins, p14, it's reptilian, p15, as the mammalian one, and basically mix and match the domains. And basically here we're looking for either a complete ablation of fusion function. And in this case, we're testing the fusion ability by incubating them on the surface of these coil muscle cells. So these cells are very readily take up the FAST protein and form these multinucleate syncytia. And what we found actually is that for the most part, most combinations of the different domains of these fusion proteins completely ablated the function. One in particular with the p14 active domain and the p15 N domain created a sort of a novel molecule with significantly higher fusion activity than either of the parents. And so we've been working sort of in this way to generate more engineered versions of fusion proteins that are sort of novel, high activity, manufacturable, and non-immunogenic. And so fast forward probably about a decade, we've optimized the PLV platform. So this is basically a novel engineered fast protein formulated with mainly neutral lipids, really with the goal to create a safe, well-tolerated, and redosable platform. Spent a lot of time optimizing, sort of started with a very complicated 14-step manufacturing process, and this has been optimized to pretty slick, continuous flow, single-step microfluidic process. And so at the same time, we've, I'll get into it a little bit as well, we've sort of reimagined sort of the lipid side of things, created panels of formulations where we can encapsulate, different cargoes like RNA or DNA or even, proteins for gene editing, for instance. I've created targeting technologies to be able to target these to any tissue in the body. And of course, even though it's a viral fusion protein, we've done a lot of work around, does this elicit its own immune response and can they be repeat dosed? So first of all, I think, looking at the challenges for the use of, traditional LNPs for systemic administration, particularly of cancer, we really focused our formulation development efforts on maximizing tolerability. And so, I'm just showing you an example here of a formulation encapsulating a DNA, so plasmid DNA that expresses EGFP, and we're putting them on a variety of different primary human cells, so fibroblasts, hepatocytes, astrocytes, and glial cells. And what we find is that if we use the formulation encapsulating DNA without any fusion protein, we can actually put a ton of them on these primary cells, completely non-toxic. So we created sort of this very well-tolerated lipid formulation. Of course, with the fusion protein, it also doesn't deliver DNA. So you see, the odd cell maybe. When we formulate this using the fusion protein, we basically see every cell taking up and expressing plasmid DNA. So we've got a sort of a base formulation that's extremely well tolerated, but can be used to encapsulate and deliver DNA in at least the primary cells. So, in our formulation development, so how do these differ than the traditional conventional LNP? So conventional LNP, just to refresh your memory, roughly half is a potent ionizable lipid. So the most common sort of base lipid is the MC3 ionizable lipid, and there's some sort of next generation ones around now that are used in our RNA vaccines, for instance. Usually about 40% cholesterol, 10% of some helper lipid that could be DSPC, and then some percentage of pegylated lipid to improve stability, sizing, et cetera. Oh, and biodistribution. And so what we find is when we incorporate these fast proteins into a lipid formulation, we actually releases a lot of these constraints. So we don't need cholesterol anymore. We actually don't even need pegylated lipid if we don't want it, although for most applications we might want it. We enable to substitute in most cases, naturally occurring biodegradable lipids that are much better tolerated. I think obviously the biggest difference is we don't need the endosomal escape mechanism. So being able to replace the, the potent traditional ionizable lipid with other lipids. And what's important here is that while we don't need the endosomal escape mechanism, we still need charge neutralization for efficient encapsulation in nucleic acid. So we basically screened libraries of ionizable and cationic lipids to find ones that had, improved encapsulation, but dramatically improved tolerability. And so, and it had the nice byproduct actually of requiring overall much less lipid in the formulation. So we need about 80% less lipid in these PLV formulations compared to LNP. And so this has had a dramatic effect on the way they behave in vivo, particularly for the delivery of DNA. And so I'll sort of give you an idea of what this looks like. So on the tolerability side, we see huge differences, and I'll show you on the biodistribution side as well. So these optimized formulations we looked at for the systemic delivery of plasmid DNA in vivo. In this experiment, we're comparing head-to-head sort of MC3-based LNP encapsulating a plasmid DNA compared to PLV encapsulated DNA. Administered systemically the LNP at 1 mg per kg and 5 mg per kg, the PLV at 5 mg per kg and 20 mg per kg. You can see here very obviously, if you look at the liver, huge differences in the way these livers look. There's pretty significant hemorrhaging, and in fact, this surprises us a bit, but both of these doses of LNP, when trying to deliver DNA, were both above the maximum tolerated dose. Every animal died. And what's interesting, the PLV, even at 20 mg per kg, these livers are completely clean. So we seem to be avoiding, whatever this toxicity is. And so I think, when we started looking at circulating inflammatory cytokines, we think this may be telling most of the story. So now we've done a second experiment where we've now lowered the LNP plasmid DNA dose down to 0.5 mg per kg. So this is down below now the maximum tolerated dose. And again, comparing PLV at 5 and 20 mg per kg. Administered systemically, looking at inflammatory cytokines at 24 hours after administration. And you can see very clearly, even at this 0.5 mg per kg dose level, when you deliver DNA with LNP, you potentiate this massive pro-inflammatory response. So pro-inflammatory cytokines like IL-6 is up 100,000-fold in the LNP group. All of these inflammatory cytokines are significantly elevated. And this seems to be particularly specific to DNA payloads in particular. And with the PLV, we actually, even in the 20 mg per kg group, we don't see a statistically significant elevation over the saline injected animals. So pretty silent when it comes to these pro-inflammatory cytokines. And we think this may be due to the route of entry. So obviously LNP are going to the endosome. Endosome is where pretty much all the detection machinery is for foreign agents, toll-like receptors integrated with the c-Gas-STING pathway. And we seem to, when we're going through direct fusion into the cytoplasm, we seem to be avoiding, the majority of these immune sensors. So that's on the tolerability side. The other big difference, when you take out cholesterol from the formulation and you change the ionizable lipid, it actually becomes far, far less liver tropic. So in this case, again, we're showing plasmid DNA, in this case, encapsulating a plasmid DNA expressing luciferase, administered at 2 mg per kg systemically. And you can see here, the liver is there. The liver gets about 8 to 9% of the injected dose, but really, because it's not getting more than that, we get pretty good biodistribution to every tissue we look at. It's also interesting as well as administered locally, in this case intramuscularly, we see good expression in the muscle fibers, good expression in the local inflammatory cells draining lymph node. We don't get systemic exposure from a localized injection. When we administer, through other novel routes like intrathecal administration for transit to the CNS and expression, we see very nice transit to the CNS and expression throughout the brain. And I guess another thing of note is that because we have this pretty big design space from which to design novel formulations, we can actually create formulations that are quite stable. In this case, formulation that can survive the GI tract intact, and this is delivering plasmid DNA orally, and we're seeing good expression throughout the GI tract. So the biodistribution of these PLVs seems to be pretty broad. We see this also in non-human primate data. We administer 1 mg per kg systemic plasmid DNA encapsulated dose into non-human primates. Again, this is a very specific formulation, very similar to the formulation we've taken to the clinic for a COVID-19 DNA vaccine. You can see here that liver is on the list, but it's about the 6th organ down. So this is not a liver tropic formulation. This is more of actually a lung tropic formulation. But we see a pretty flat biodistribution throughout all organs. And you can see here there's a variety, pancreas is there, there's heart, there's bone marrow, muscles there. So we're hitting a lot of different organs with these PLVs after systemic administration. So obviously the PLV platform is a combination of lipids and a viral fusion protein. And you might expect that a viral fusion protein could be immunogenic. So we've done a lot of work around, trying to sort this out. We were enthusiastic sort of about the fact that Roy took a long time to elicit monoclonals or polyclonals against the FAST protein. It seems to be pretty immune silent, and this is borne out by this data, sort of a snippet of what we've done. In this case, we're delivering RNA encapsulated in a PLV, either delivered intramuscularly or systemically. and we're doing repeat dosing, dosing once a month for 5 months. And then we have a GLP validated, sort of clinical anti-FAST protein ELISA, and basically nominal response. So a couple animals in the IV group just above the lower limit of detection, pretty nominal. We get actually good antibodies against luciferase, which is really interesting. We've also done this in non-human primate. This is one example where we dosed 11 subjects with a pretty whopping dose of DNA. So 6 mg per kg, just to put into perspective, I think it's more DNA than the monkey had to start with. We've given them 2 doses of 6 mg per kg, spaced a week apart, and then collected serum on day 17, tested for anti-FAST protein antibody by our validated ELISA. And again, very similar story, nominal response, couple animals just above that lower limit of detection. So, so despite the fact this is a viral fusion protein, we seem to be able to repeat dose effectively. And, we're all in the nanotechnology field, so we're aware of this. I think tolerability toxicity has been a challenge for many different companies and technologies as they bring their candidates through GLP tox and clinical trials. And so we spent a lot of time on large animal tox. We've administered PLVs to primates and canines, 6, 10, 20 mg per kg, single dose, multiple dosing. I would say the key difference that we see between PLV and LNP is we don't see any histological changes in any of the major uptake organs. We don't see any significant immune toxicity. And so maybe a bit of a bump after infusion that's resolved after 24 hours. And we've done a nice clean GLP tox study in rat for our COVID-19 vaccine program. And one thing that this group may be familiar with is CARPA, the complement, sort of this complement-mediated hypersensitivity response. You can get to nanoparticles. And so there are assays available to sort of test this in vitro, and so we've optimized our formulations to have as low reactivity as reasonable for CARPA as well. And of course, we have a clinical program, a COVID-19 DNA vaccine that began at the beginning of the pandemic. We've now taken that to the end of phase 2. We're hoping soon to be able to start some phase 3 booster activities in Canada in the short term. But basically, this is a DNA-based, intramuscularly injected vaccine expressing, a full-length spike with a couple of DNA-based genetic adjuvants, and so we're actually really excited about this platform to be able to create a sort of a nanoparticle-based DNA vaccine platform. We actually see. In our preclinical studies, we see very, very potent responses at very low doses. You can see here, even at 1 microgram of DNA, we're getting some pretty good responders, and 10 micrograms, we're getting full response in all the animals, both IgG and neutralizing antibodies. And so very excited about taking this platform through, hopefully, someday to approval. But I just wanted to highlight that, and that, we've taken this to clinic now. We've had a total of 268 individuals exposed to the platform, very, very clean safety so far. So we're excited about, utilizing this and moving on to things like cancer. And so with that, let me talk about, I'm going to spend the rest of my talk discussing our efforts to develop a novel suicide gene therapy approach to cancer. And as I alluded to at the beginning, really, and alluded to, at the beginning is that, really the goal for us is to create something exquisitely selective so that we can ideally avoid all of the side effects that are traditionally associated with cancer treatment. And so the idea here is that, so what I've shown you is that the development of a platform where we can encapsulate and deliver DNA. The great thing about DNA, maybe compared to RNA, for instance, is that they incorporate very selective promoters. And these promoters can drive actually pretty complex, I would say, DNA programs written sort of in a plasmid. And so the idea here is we can utilize both the selectivity of the PLV, which I've shown you is actually not very selective, it goes everywhere, but really drive the selectivity through the selection of a cargo. And so the idea here is that we want to selectively kill the tumor cells. So I like to say based on what they're thinking. So the idea here is that tumors acquire different mutations as they evolve. These mutations really activate oncogenic pathways that aren't activated in many other cells. These oncogenic pathways can be utilized to basically drive selectivity of a pro-killing based DNA program. So the idea here is we use a tumor-specific promoter we use this really interesting molecule called ICAS9. So it's an inducible caspase-9 that is expressed selectively only in tumor cells. So we're basically delivering this all over the body. It's getting into every cell of the body, including the tumor. In the tumor-specific promoter drives the expression of this ICAS9. ICAS9 by itself doesn't have any effect. When you administer a small molecule dimerizer, in this case, we have our dimerizer is rapamycin, which is a well-known, clinically approved molecule. It dimerizes the ICAS9 and basically has recruited the apoptosome and drives really potent tumor cell death. So the idea here is that you're loading yourself up with nanoparticles. They're only expressed in the tumor, and then you're taking rapamycin to activate the dimerization, which kills only the tumor cells and doesn't have any effect on any other cells. What's nice about this approach, in my mind, is that, it doesn't distinguish between primary site and metastasis. This is traditionally, metastases are very difficult to treat with many therapies, and ideally very selective for tumor cells. And so we actually chose the p53 pathway. So we have a number of different ones, but I just wanted to show you data today on p53. Obviously, p53 is the guardian of the genome, implicated in pretty much every single hallmark of cancer, and upstream of many different pathways that are relevant for the growth and survival of cancer. And if we sort of take this out of if p53, so cancers basically evolved to escape the effects of p53, which again has an effect on pretty much every single aspect of the way cancers grow, survive, and proliferate. And so we thought, so if cancers are actively mutating or ablating p53 function, what is this doing to the upstream signaling of p53? So obviously p53 is needed, to regulate sort of sense and provide effector function upon damage, for instance, in the cell or stress in the cell. So if you actually mutate or take away that function or ablate p53 altogether, you might expect that the transcriptional activation of p53 may be ramped up, right? Because this feedback loop. And we actually see this evidence for this in sequencing studies. If we look at sort of normal tissue compared to cancerous tissue in a wide variety of different cancers, and now you're looking only at p53 transcriptional activity. So again, even if p53 gene is completely ablated, gone, the activation signal to turn on p53 is still there, and in pretty much every case we looked at, significantly elevated. So p53 function or the gene itself is gone, and the cell is just trying to hit that button, trying to turn it on. So you can see the activation signals are dramatically ranked up in all the cancers that we look at, and we validated this by in a human cell line by basically ablating, so using gene editing to ablate p53. And we can see when we ablate the p53 gene, there's absolutely no expression of the protein. We see using a p53 promoter reporter driving luciferase, we can see huge upregulation of the activation signal for p53. So we thought, what if we hijacked this highly active p53 activation signal to selectively target and kill those cells? And so the first thing we did is we looked at, okay, so this kind of makes sense. We see it in human tissue. What if we look at a wide variety of different cancer cell lines and compare those to, and these have, sort of pretty much every different p53 modification under the sun, mutations, deletions, et cetera. And I compare these to basically normal functional p53 cells, and we see huge activation of the p53 transcriptional activation pathway. So anywhere from about 10x to 3 or 4 orders of magnitude. So this seems to track. And again, so just to recap, the idea here is to hijack pretty much any dysfunctional p53 transcriptional activation to selectively target these particular cancer cells and deliver a payload that will basically just politely ask them to commit suicide. So through this inducible caspase-9, have these cells go through selective apoptosis. And so we first tried this in vitro. So in this sort of pretty straightforward experiment, we took an engineered p53 promoter driving AdCas9 with the GFP as well. We mixed so H1299 lung cancer cells, which have an activated p53 pathway, with IMR90 fibroblasts, which have intact p53. We co-cultured them, transfected them with the construct, and then added rapamycin to induce dimerization of the AdCas9. You can see very clearly here the GFP positive cells at the beginning of the movie, basically all very, very quickly go through apoptosis. All of the red cells, which are the IMR90s, are completely left intact. So very selective approach to kill the cancer cells. And so obviously we think they're going through apoptosis, but we wanted to verify this experimentally. So again, when you add rapamycin, so when you transfect cells with this p53 AdCas9, add rapamycin, you see very, very clearly the induction of proapoptotic markers in H1299 cells. So these constructs are definitely inducing apoptosis. And so how does this, so I told you at the beginning, the idea here is to basically systemically administer plasmid DNA. It goes everywhere. But we activate that only in tumors. And so this is an experiment that really validated this hypothesis for us. So in this case, we're administering a reporter gene, and the reporter is either CMV, so ubiquitous promoter, CMV driving luciferase, or our engineered p53 promoter driving luciferase administered systemically. And you can see very clearly when you administer systemically with a CMV promoter, you see expression everywhere. So every organ we look at is expressed in luciferase. If you administer, basically the same PLV, very similar plasmid. In this case, the only difference is that the promoter is p53, and so we know that this plasmid in all these organs, we see zero expression in any organs. In this case, this is a tumor-bearing mouse, and we can see very clearly here that the selective expression of luciferase only in the tumor and basically nowhere else. So we did sort of we got really excited about it. So this is positive. So we started out with just a very, very simple experiment in prostate cancer. I'm just showing you one animal. We did this on tens of animals. But basically, in this case, we grew xenograft PC3 tumors in the flank of mice, immunocompromised mice. We injected, in this case, we did an intratumoral injection of the PLV with the suicide gene payload. And then we activated with rapamycin. You can see here very, very quick ablation of that tumor. You see about 90% reduction in solid tumor mass after just a couple of days with this. So this really validated for us that this could work in vivo. And then we've done quite a number of experiments since then to look at sort of the kinetics and the efficacy of the suicide gene therapy in different tumor models. In this case, in an immunocompromised tumor model, this is H1299 again. We're sort of doing a dose range finding study. In this case, looking at when we inject the PLV, when we inject the rapamycin. We can see here, very clearly after a few days of treatment, we get good statistically significant reduction in tumor mass between the non-treated and treated groups. And so this is an immunocompromised model. So we thought that obviously because we're causing cell death, we may be potentiating an immune response as well. I guess first of all, so I guess the one big question when you're administering an nanoparticle systemically and looking for uptake in tumors, how's the distribution within the tumor? Because tumors can be highly heterogeneous with sort of differential vascularization, for instance. We actually were gratified to see we saw pretty good. In this case, we're looking at tunnel staining in these tumors. We're seeing good, tunnel staining throughout the tumor mass in these tumors as well. But we were really interested in taking this to an immunocompetent model. This is an example of an experiment we did in a CT26 colon carcinoma model. In this case, we were looking at either single agent treatment with the p53 driven AdCas9. We called this, our code name is OSX001. It was dosed twice weekly at 12 mg per kg. And then with or without checkpoint inhibitor, in this case, anti-CTLA4. And we actually saw some pretty dramatic responses. So single agent treatment group with OSX001 was tumor growth inhibition of almost 94%. When we combined the two treatments, we saw almost 100%, so getting up there to almost 98%. And again, in this case, this is not necessarily the most immunologically hot tumor, so the checkpoint inhibitor dose that we used only produced an 80% tumor growth inhibition. What's interesting is when we combine the two, we saw complete regression of tumors. What's also interesting is we seem to be inducing a potent and durable response. So if we then took these animals that had full tumor regression, so the combination treated animals that were treated with both OSX-001 and the checkpoint inhibitor, and then re-challenged them 70 days later with a second CT26 tumor of the same starting size. So basically repeating the same experiment on these cured animals, we actually saw complete response. So these tumors were not able to grow compared to control. And so what's nice is we're producing a durable anti-tumor response, which is very encouraging. So we went to some more challenging models. So the V16F10 lung metastatic model is a very, very challenging model. You inject, so these are melanoma cells that are injected IV in animals. They transit many places, including the lung. The lung's sort of easy to look at, but these animals get sick very quickly and they die within about a week. So if you have a response in these tumors, it's a good thing. And so we looked at basically the same kind of experiment where we looked at either single agent treatment with the p53-driven ICAS9 activated with rapamycin, which we saw a good response, or combined with checkpoint inhibitor. If you look at sort of the quantitative data, the checkpoint inhibitor combination did a little bit better, but we saw a pretty good response with a single agent treatment. And what was nice as well, we asked, so this is a very exciting response from this sort of challenging model. We started to dig deeper into because the idea here is that obviously through caspase-9-based apoptosis, I wouldn't say it's really designed not to be too much of an immunogenic type of cell death. So are we still, is the ICAS9 potentiating an immune response that is synergistic with CTLA-4 treatment? And we seem to see that. So when we look at the Treg population, we see a significant reduction in Tregs, particularly in the combo group. We also generated an assay where we basically took the splenocytes of treated animals and then incubated these with p16F10 cells to look at the potentiated cell killing. And we can see, very clearly animals treated with either the p53-targeted PLV or the combination of that and the CTLA-4 inhibitor. We're seeing a very sort of selective and potent cytotoxic T lymphocyte response. So it seems like there is some immunogenic cell death. And so for us, we're looking now at potentially alternative payloads that potentially sort of more of an immunogenic cell death to see if we can move the needle on this a little bit more. So I think at this point I'll summarize and happy to take questions. So what we see, so we've generated basically a proteolipid vehicle platform that enables the systemic delivery of DNA. And that systemic delivery of DNA allows us to create some really selective cargoes that are basically delivered everywhere, but selectively activated in cancer cells. In particular, the engineered p53 promoter that we're using is pretty dramatically upregulated in every cancer cell that we looked at. And it seems that if we're using a DNA vector, using this p53 targeted approach, seems to be a pretty effective means to selectively target these cancer cells. And we found this both in vitro and in vivo. And somewhat surprisingly, so obviously we saw some synergy with an immune checkpoint inhibitor. We did seem to be potentiating at least some immunogenic cell death in this model to really see a synergistic effect with the checkpoint inhibitors. And I think, next steps for us are really to, try to increase the immunogenic cell death by potentially driving other cell death pathways. So I'll end it there. There's obviously a great number of people that contribute to this project, people in Oncocenix, people in my academic lab, Roy Duncan's lab, really the grad student in my lab, Doug Brown, really did most of the work that I presented today. just a fantastic driver of this research. And I'll mention as well, this OSX001 is being commercialized by a spin-off company called Efficacy. Excited, actually, at the beginning of the pandemic to bring this in first-in-human trials in Edmonton, Alberta. We're sort of derailed by the pandemic, but now that we've taken the platform to clinic, we're really excited about working with this team to bring FAST-LNP therapy to cancer patients. Thanks so much for your attention, and happy to take any questions.
As I was thinking about this, this viral targeting approach to enhance the fusogenic property, and I was also thinking, you were asking about, how to induce immunogenic cell death as well. I'm wondering, viruses use things like neuraminases and so forth to chew up sugars that are on the surface. Are there ways for you to modulate sugars to enhance this as well?
Yeah, that's a really good question. So we actually haven't looked at sugar specifically. What I do know is that, so the FAST proteins so they don't have a ligand, so it's purely a biophysical interaction. So they just need to get close to the target cell to induce fusion. But obviously the sugar, sort of like a PLX, whatever the cell is, could have an impact on its ability to do that. Now we have been looking at other mechanisms of cell death, like necroptosis, for instance. I think, we've seen some pretty good early data there. So I think that's definitely the direction we're going.
Cool. Is there an ideal size for these liposomes, or I'm trying to think about the sort of the mechanism of fusogenic activity?
Yeah, so I would say pretty much exclusively smaller is better. So we spend a lot of time optimizing the manufacturing process to get smaller particles. I think, for any systemic delivery, you want under 100 nanometers. If you want, proper biodistribution, but the lower we can get, we seem to get better tissue penetration, and particularly in tumors.
Right. I'll just wait for a minute. I have a bad habit of over-speaking people. So let me see if there's a question popping up. Not at the moment. You had one example where you were getting exceptional delivery to lungs, if I remember correctly. It was one of the first ones that you showed, one of the first slides. And I'm just curious, what was it? Do you know what the reason was you were getting such good delivery to lungs?
Yeah, that's a great question. So, so I didn't really get into it. So we really have two mechanisms of targeting different tissues using the platform. So we can use ligand-mediated, interactions. For us, this is actually the least preferred approach, because if you introduce ligands, then you have to worry about, their degradation, and manufacturing becomes more complicated. Again, because we have this pretty big design space as far as the lipids go, we much prefer working with different lipid ratios, different interesting helper lipids, different interesting ionizable lipids, and then playing around with the surface charge and other things. And so we've actually worked to create formulations that target muscle, the target lung, the target CNS, the target kidney, and I'm not going to answer your question specifically. So I think in that particular experiment, we used for the systemic administration in primates, we actually used our vaccine formulation. The average size of that formulation is in the 100-120 range. And I think so that's the basis of its lung tropism. It's basically the first organ it's seeing. But we do have other formulations that are smaller size that are also lung tropic, and that's driven by the inclusion of very specific lipids.
How was that delivered? How was that therapy delivered? Was it IV injection? Where?
Yeah, they're all IV systemic administration.
Right, but where? I mean, so sometimes if you want to, get to the lungs, you can actually use an artery up here somewhere, or a vein up here. I guess artery up here somewhere.
Correct. Yes.
Yeah.
So that's how that experiment was done, where we saw the lung tropism.
I see. Okay, got it. Okay, got it. Do you see differential loading capacity with the shorter nucleic acids compared to the longer ones? This is, a lot of design had to go around that, or some design had to go around that in the ionizable lipid platform. How did you guys have to confront this at all, or how?
Yeah, absolutely. Yeah. So I mean, we use microfluidics. So obviously platforms like Precision Nanosystems and the nice thing I like about these is they're self-assembled. So there's really no limit on the size of cargo you can put in it, but you do have to have a lot of thought around, N to P ratio, et cetera, and the different lipids that go into it, because they can have a dramatic effect on encapsulation efficiency. And so, yeah, so I'd say generally shorter things are easier to encapsulate, but I guess with the right ratios, larger things are straightforward as well.
Let me just pause here for a second. I'm so sorry to keep on interrupting. Is there any, I know that it seems as though these fuse quite rapidly. And I'm wondering, and you don't have very much lettuce around the outside of your liposome. And I was thinking about the classic example of adding PEG and so forth to increase circulation time and so forth. But I didn't remember seeing a PEG in the lipid that you had, right? So that means, was there? Maybe I missed. I'm sorry if I missed, in fact.
Yeah, so typically for systemic administration, PEG or some sort of shielding polymer is required. Yeah.
Okay.
Yeah, and then it's a nanoparticle, right? So the half-life in the circulation is not going to be more than 48 hours, for instance.
Okay, and then one last question that I have off the top of my head. You had an example very close to the end of your seminar where you talked about reintroducing an organ and seeing whether the new introduced organ would grow. And I was trying to understand what the residual drug was that would have persisted, I guess, long enough to affect the incoming organ. I didn't catch what would have persisted.
Sure. Yeah. So that experiment is specifically testing the induction of a specific anti-tumor immune response. So you basically take these animals, you put the tumor in, and then you're creating an immunogenic cell death that actually creates specific cytotoxic T cell populations in the animal that will then surveil and get rid of any tumor cells.
Got it. That was the way that worked. Okay, thanks very much. I'm going to open the floor to more questions. Diana?
John, did I hear you say that following administration that the immune system learns to recognize these cold tumor cells more effectively, and there's some sort of longer term preventive feature? And is that because you've trained the immune system, or the process causes the tumor cells to create neoantigens that allow them to be recognized?
Yeah, that's a great question. So in this particular circumstance, these CT26 tumors have sort of well-known characterizable neoantigens. And so we're creating, basically through our treatment, we're creating enough immunogenic cell death to show the immune system those neoantigens, and we can actually measure that specific response. I think, in humans it's a little bit more challenging because every tumor is different. But it's the idea of creating these, personalized cancer vaccines as well.
And have you been able to study how long that learning curve lasts? Is it a permanent learning curve, or is it time limited?
Yeah, so I think, there can be very, very long-lived T cell populations. So I think if you can induce that potent response and get long-lived, T cell cytotoxic T cell populations, I think it should be durable.
Okay, that's really interesting.
I see that there is a question. Do you have data on anti-FAST protein antibody generation in human?
Yeah, that's a great question. So as I mentioned, we have a GLP anti-FAST protein ELISA that we developed for a clinical study. We've administered our vaccine to 268 individuals and tested all of them with the assay, and no anti-FAST protein antibodies that we were able to detect. I'll just mention, I guess this is an intramuscular injection of a maximum 250 microgram dose. So that might change if we're doing systemic administration of larger doses.
Got it. Thank you. The next question, I guess, another anonymous attendee, so I'll have to ask it myself. And the question is, 20 milligrams per kg delivery of PLVs resulted in paler than usual liver, while higher doses of LNP exhibited liver damage of the ionizable lipid. Is the paler liver not a symptom of fatty liver, or was it just the lighting that made it look like that?
Yeah, that's a great question. So I think those images were taken 24 hours after administration. So I don't think that's fatty liver. I think it's just the lighting. From a pathological standpoint, we had an independent pathologist look at actually the NHP study, 20 mg per kg versus saline injected animals, and wasn't able to tell the difference.
Okay, very good.